Welcome dear learners. Today we are going to discuss about layers of DNA replication. The content and outcome of this today's presentation is, after listening to this presentation, you will be able to understand different enzymes involved in DNA replication and significance of this enzyme in DNA replication. Introduction. Multiple proteins are required for DNA replication at a replication fork. This includes the DNA polymerase, the major enzyme which is involved in DNA replication. Along with that, it requires several other enzymes, namely helicases, which unwinds the DNA, primase, which synthesizes a small stretch of RNA primer, topoisomerase, which is involved in relieving the torsional stress in the DNA, and DNA ligase, which seals the gap. Apart from this, there are several other enzymes which we are going to discuss today. Most of the enzymes, they are multi-subunit protein complexes. DNA A protein. The base sequence at the origin of DNA replication is recognized and bound by the DNA A protein. DNA A protein is the circular DNA A protein, which is the first protein to interact with the DNA and initiate the process of DNA synthesis. It identifies certain 9-mer and 13-mer sequences which are present upstream to the DNA replication. Here we can see when this protein binds this close complex, that is the two strands of DNA, they are held together in double stranded structure. They are converted here into single stranded structure, that is a replication bubble has been formed. So this is because of the presence of DNA A enzyme. Now this DNA A, it binds cooperatively. When it binds, it immediately is followed upon by DNA C protein and DNA B protein. DNA B protein is also known by another name called as helicase. DNA B protein or helicase, it uses energy from ATP to break the hydrogen bonds which holds the base pairs together. Mind you, the two strands of DNA are attached with one another by complementary base pairing, and in that base pairing the hydrogen bonds play important role. This bonding are broken by enzyme called as DNA B protein or helicase. This allows the two parental strands of DNA to begin unwinding and forms two replication forks. Next, SSB protein. Single-stranded DNA binding protein, or in short we call it as SSB protein. It binds to the single-stranded portion of each DNA strands, preventing the strands from reassociating and protecting them from degradation by nucleases. Mind you, when the two strands are separated, they are liable to be exposed to different types of enzymes. So to protect them from such enzymes, they are immediately covered upon by enzymes called as SSB. This property helps promote the denaturation of duplex DNA. In eukaryotic cell, replication factor A serves the role of SSB. So in short we also call it as an RCA, replication factor A. Next enzyme, primase. Primase is an enzyme that copies a DNA template strand by making an RNA strand complementary to it. So primase, it synthesizes a short stretch of say around 10 nucleotides RNA primer in the 5' to 3' direction. The parental strand is used as a template for this purpose. The RNA primer are required because the DNA polymerase are not able, here we can see this, they are not able, this is a very distinguishing point we are going to discuss, that is, in case with replication it cannot start the de novo synthesis. For that it requires a short stretch of RNA primer. So RNA primer are required because DNA polymerase are not able to initiate the synthesis of DNA on their own, but can only extend a strand from the 3' end of preformed primer. The enzyme is active only in presence of other enzymes, that is helicase, and it creates a complex called as primosome complex. So it keeps a track, or it checks that this is used only during the process of DNA synthesis. Next, DNA polymerases. There are several different types of DNA polymerases. In E. coli, at least 5 different types of DNA polymerases are known. All of them are capable of DNA synthesis, but not all of them are used all the time. DNA polymerase third is regarded as the major replicative enzyme. Major replicative enzyme means it is mainly involved in the process of DNA replication, while the other, they substitutes the function and they are used for some other task. So why DNA polymerase third is regarded as the major replicative enzyme and not the others? Because of two reasons. The DNA polymerase third is one of the processive enzyme. It is very fast. It attaches around about 1000 nucleotides per second, and it is also fiddle, means once the enzyme it bounds to the DNA, it completes a long stretch of DNA replication before getting dissociated. Now it catalyzes the chemical reaction for polymerization of nucleotide. DNA polymerase third begins the synthesis in 5' to 3' direction. Note again, this is very important and vital point. No DNA polymerase is available in nature which can synthesized in 3' to 5' direction. So all the polymerases that are present, they can synthesized only in 5' to 3' direction. DNA synthesis. The newly synthesized strand is complementary and anti-parallel to the parental strand used as a template. Next, DNA polymerase first and RNA is H. Mind you, this A should be small a. This is an enzyme. As the name suggests, RNase H, it cleaves the hybrid DNA RNA hybrid and is involved in removal of primer during the process of post replication after the synthesis of DNA. Removing RNA primer in the processing of DNA after replication. This enzyme removes the ribonucleotide one at a time. That is, it shows exonuclease activity and it removes one nucleotide at a time. Unlike we have discussed in DNA polymerase, where it was able to synthesize 1000 nucleotide per second. So here it removes one nucleotide at a time. DNA polymerase first also fills in the resulting gap by synthesizing the DNA. As we have discussed, all the DNA polymerases are capable of DNA synthesis. So by removing the RNA segment, the gap which was formed, it is immediately filled by DNA polymerase first. Both DNA polymerase first and third have the ability to proofread. Now this is again very important characteristic possessed by DNA polymerases. So to proofread means whatever nucleotide they have attached, they can go back and remove them. Suppose during addition of nucleotide, the complementary base pairing has, the order has been lost, or any miss nucleotide have been incorporated over there. So immediately the DNA polymerase goes back and removes that nucleotide. If DNA polymerase makes a mistake during the DNA synthesis, resulting unpaired base at the 3' end of the growing strand is removed before the synthesis continues. So mind you, this is very vital because DNA carries the genetic material. It is the blueprint of information, genetic information, and any such change or any such corrections, if kept unchecked, it can lead to a loss in genetic. It can lead to a vital loss to the coming cell or next generation. Now, comparison of DNA and RNA polymerase. DNA polymerase which is involved in replication event, RNA polymerase which is involved in transcription process. So nucleic acid synthesized in case with the DNA polymerase or replication, it is DNA. So from DNA is synthesized. While in case for transcription, from DNA, RNA is synthesized. So the required template for this is the DNA molecule. Here again, in transcription, it also requires the DNA as a template. The required substrate for DNA replication, it requires DNTPs. Mind you, this is the difference between NTPs and DNTPs. DNTPs means deoxyribonucleotides, where in transcription oxygenated, that is 2' 3' oxygenated nucleotides are attached. Primer is required in case with replication and it is not required in case with transcription. Proofreading, yes, it is very vital because it carries genetic information. The proofreading activity is must. Clamp and clamp loaders. Protein from the DNA polymerase third holoenzyme complex holds the DNA polymerase to the RNA. Holoenzyme means a complete enzyme. This helps the DNA polymerase complex to stay to a DNA and through an entire cycle of replication. Mind you, DNA is very thin or it is a thread-like structure, and these enzymes, they are comparatively very larger proteins. So for the process of replication to be completed till the end, this has to be remain in association with the DNA molecule. For that, this clamp loaders plays vital role. Multi-subunit enzyme called the complex function as clamp loader. That is, it loads the clamp on the surface of the DNA. In eukaryotic cell, multi-subunit protein called replication factor C, RFC, it serves the function of clamp loader. Next enzyme, DNA ligase. DNA ligase seals the nick between the Okazaki fragments, converting them to a continuous stretch or strand of DNA. Now covalently closed nick in double-stranded DNA synthesis. Look here, as we have discussed, there is no enzyme, there is no DNA polymerase which can synthesize from 3' to 5' direction. So this gap has to be filled up only with the help of an special enzyme which can do the task, that is DNA ligase. DNA gyrase. Next enzyme, DNA gyrase. As helicase unwinds the DNA at the replication fork, the DNA ahead of it becomes ovobound and positive supercoil forms. The DNA gyrase inserts the negative supercoiling by nicking both the strands of DNA. Mind you, DNA gyrase is a type of topoisomerase second which nicks both the strands, means double stranded cut, passing the DNA strand through the nick and then resealing both the strands again. Here is a list of all the enzymes which we have discussed, and there are different types of genes from where they are synthesized and the functions they execute. So in summary, what we have discussed in this presentation, the enzyme or proteins and their functions. DNA A is the first protein to bind to the DNA and initiate the DNA replication. DNA B, called as helicase, it unwinds the DNA strand. SSB, it binds to the single-stranded DNA structure, protects it from the attack of nucleases. Primase, it is involved in the synthesis of a primer. DNA polymerase, actually involved in the DNA replication, it forms the phosphodiester bond. DNA ligase, it seals the gap. Topoisomerase, it maintains the torsional stress and imparts the negative supercoiling. So for this presentation we have referred two books, Molecular Biology by David Clark and Gene 7 by Benjamin Levin. Thank you.